Maxim and Gilbert method of DNA sequencing. This method of DNA sequencing, it makes use of chemical reagents to bring about base-specific cleavage. Although this method it was used in mid-70s, and later on it was replaced with more suited methods that was based on enzymatic reaction, but still we can discuss this method so that one can get basic information from the history that how DNA sequencing first it was started. And as I told you that this method was developed by Maxim and Gilbert around 1977, and although this was soon replaced by another advanced method. In Maxim and Gilbert method, the starting DNA fragment, it may be restriction DNA fragment or any target DNA which we want to sequence. And the target sequence DNA fragments, first they are labeled with radioactive material. Rather they are AND-labeled, like phosphorus 32. It is a type of radioisotope of phosphate that can be used for AND-labeling of DNA fragment. And in this case, the base-specific cleavage by different chemical reagents, it is brought about in 3 basic steps. In the first step, the DNA fragments that are AND-labeled, they are treated with certain chemical reagents that can alter 1 or 2 bases within the DNA fragments. मिसाल के तौर पे जो एक chemical है which is called as dimethyl sulfate, it can methylate guanine base at N7 position, so that it is now altered or modified, and once it is modified, the DNA elongation during the course of sequencing, it cannot proceed further. So then the altered base in the next step, it is removed by using certain chemicals from the sugar backbone, and after that the removal of the altered base, then the sugar bond it can be cleaved. So in this way we will get a DNA fragments whose elongation has been terminated. Why? Because of the presence of some modified nucleotide bases. In Maxim and Gilbert method, हम जो है बेसिकली 4 या 5 रिएक्शन परफॉर्म करते हैं हर बेस के लिए अलग अलग, ठीक है. तो इसमें जो डिफरेंट जो रिएक्शन मिक्सचर हैं इनको डिफरेंट एपेंडोव्स या टेस्ट ट्यूब में परफॉर्म किया जा सकता है. फॉर एग्जांपल, फॉर guanine there may be a 1 reaction mixture, and this base it can be altered by its respective chemical reagent. Then in the next reaction mixture, it may be for suppose adenine, its alteration it can be mediated by another chemical reagent, and similar is for thymine and cytosine. So we may have different type of reaction mixture for different bases, so that when we will use AND-labeled DNA fragments, the elongation it is carried out for limited period of time, because whenever modified base is inserted, the elongation it is stopped, and then 1 or 2 cleavages they are introduced per DNA fragment. So in this way, when we complete the reaction in different reaction mixture, we will get mixture of fragments that may be variable in length, and these mixture they are then separated by using polyacrylamide gel electrophoresis. I think you still have the idea that we discussed that either agarose gel or polyacrylamide gel, it is used for the separation of DNA molecules depending upon the fragment length. So similarly here, after performing the reaction, the different fragments of the DNA, they can also be separated by polyacrylamide gel electrophoresis, and percentage it may be 6 to 20%, and in the presence of 7 molar urea, that will prevent the formation of secondary structure of the DNA. And even we can also apply high voltage around maybe 70 volts, so that the temperature it will high, and it will also prevent the formation of secondary structure. When gel run is complete, then the autoradiographic technique, it can be used to detect DNA fragments. So chemical targets in Maxim and Gilbert DNA sequencing strategy, it is shown here. And here it is the dimethyl sulfate, first chemical reagent that can attack adenine or even guanine, which may be purine bases, and hydrazine chemical, it can attack cytosine or thymine. So different chemical reagents, they can alter the different set of nucleotide bases. Again, this table indicate different set of bases and chemicals that can alter nucleotide bases. For example, in case of guanine, the chemical reagent, it is dimethyl sulfate. As I told you that when base is altered, then the altered base, it can be removed by piperidine. And finally, the sugar bond, it again can be cleaved by piperidine, so that we will get a short fragment of DNA. And similarly, guanine and adenine, again it can be altered by using certain acids. Thymine and cytosine, it is again can be altered by hydrazine. Cytosine by using hydrazine and NaCl. And adenine or cytosine, again it can be NaOH. So here we have 5 different type of reaction mixture that are run separately, and depending upon the alteration of the base pair, different fragments that vary in their length, they will be obtained, that will be separated by using polyacrylamide gel electrophoresis. And here it is the basic strategy of Maxim and Gilbert method. सपोज करें कि ये हमारे पास डीएनए का फ्रेगमेंट है जिसका सीक्वेंस हम जानना चाहते हैं. Now the base specific cleavage is there. Suppose here we have 2 at 5' end cytosine bases, and if they are altered in the reaction, so the shortest fragment that we will get, it belongs to cytosine and cytosine here, and next base is guanine, and here the base specific cleavage of guanine, it again will produce a short DNA fragments. So in this way, if we will move on in different reaction mixture, we will get fragments of DNA that will vary in length, and that will be separated on polyacrylamide gel electrophoresis. And here we can know the sequence of the DNA in the reaction mixture. So the different fragments that are variable in length, they will indicate the sequence of the DNA fragment. So this is the basic strategy of Maxim and Gilbert method, and it is a type of manual sequencing scheme.